Well, thank you. Thanks for that introduction. So I'd like to keep this as informal as possible, and I'm actually going to start by asking a question. So I work on infectious diseases, and I just want to get a sense from the audience, how important do you think infectious diseases are in the world in terms of the numbers of deaths? That's something that we can measure.
Huge.
Huge. Okay, so what percentage? A fifth, okay, a fifth, a seventh,
Half.
a third, half.
Two-thirds?
Two-thirds, okay, this is, well, you know, this is good. All right, so here is the data this is from the World Health Report. I think this is the reporting the 2003. So it's about a third of deaths. So whoever got third, there we go, that person gets the first star of the day. Okay, so, and I think this is very important. You know, I speak to many audiences about this, and it's surprising how the range of numbers, just like we saw here. So that, and this figure probably hasn't changed very much over the last 100 years. Now, why might that be? Why are infectious diseases still a problem? You know, you think about this, you know, we'll talk about the major infectious diseases. Maybe we should talk about those next. What are the major infectious diseases in the world?
Tuberculosis.
Tuberculosis,
Cholera,
diarrhea, HIV,
malaria.
Diarrhea, malaria. Good, yes. Okay, so this is a great audience. So this is actually from an op-ed piece from the New York Times, written at about the time of the Sars epidemic. It's April 30th, 2003, by Howard Markle and Stephen Doyle. And this is presented in this kind of banner format, and the size of the banner actually indicates the burden of disease. And the way we measure burden of disease, there are many ways to do it, but kind of the most generally accepted way is deaths plus disability induced by the disease. So you can see, you were absolutely right. Tuberculosis, in fact, is the most important disease from the point of view of burden. And we'll talk just a little bit about tuberculosis. Malaria is clearly very important. This is in 2003, so the HIV/AIDS banner has probably grown somewhat. This is one you missed, hepatitis B. This is a very important debilitating disease. Here are diarrheal diseases. And this just tried to put SARS in perspective relative to some of the other major threats. Not to say that any disease is unimportant, but just to give a perspective of the burden of disease. Okay, so I'm going to talk just a little bit generally about infectious diseases, and I'm going to focus on malaria, which is the disease I'm most interested in and spend most of my time working on. But I want to give you a sense kind of how I like to think about diseases and think particularly about infectious diseases. So why is it? I mean, think about this. Tuberculosis has been around, and we'll talk about the history of tuberculosis and malaria for a long time. AIDS actually and SARS are relatively recent entries into human health, but TB and malaria have been around for years. Diarrheal diseases. Why don't we have a cure? Why haven't we solved this problem? Yeah?
I think at least for malaria, it's really been neglected.
Okay, neglect is certainly something, lack of awareness, you know, in places where there's lots of money, okay?
AIDS, I think, because it's an RNA virus, and RNA has a high rate of mutation and replication. And
okay, so AIDS evades the immune response because of RNA virus. There's another.
I mean, more looking to the future, the fact that they're evolving the resistant strains.
Right, so resistance is a big problem.
But I don't think it was until recently, though, so it's kind of hard to...
Well, okay, but resistance, you know, we've had drugs for TB and malaria for the last, you know, at least century or late last 100 years, so, you know, okay, out there.
I think sometimes some of them affect individuals in the third world, so I think that, I don't know, maybe I'm...
Right, no, exactly.
Either we just haven't put, if they affected us, we put more of a focus on it.
Right, that's right. Some of it is just lack of focus, right? No, that's a very important point. Okay, other ideas?
I know conditions in many areas have gotten worse, like TB in Russian prisons.
Right, so there are issues of transmission or spread, right? In the case of malaria, you have global warming, you have mosquitoes populating more of the geographic area of parts of the world. Okay, it's a good group. We can leave out the slides. Okay, so we have been successful, though, Okay, some things, right? Look at this, we've got antibiotics. Those were developed for infectious diseases. We've completely eliminated many diseases. Vaccines, think about it, polio. None of you are probably really old enough to remember polio. Okay, all right, so polio is essentially gone from this hemisphere, right? So it is possible to overcome infectious diseases, and leprosy, for example, is almost eliminated by drugs. It's a very similar organism to TB. It's interesting why they're not completely eliminated. So here are the reasons, we've gotten them all, I think. Why do we still have infectious diseases? There's the globalization of risk. We talked about that a little bit, this issue of spread, and we'll talk about that a little bit more. Drug resistance, there are few new drugs, and there are also new diseases. This is one that we missed, right? And we're going to talk about new diseases a little bit at the end, because I think it's also very important as we think about infectious diseases. This isn't a static thing. And one of you said evolution, selection of resistance is very important, but actually selection of new agents into humans, right? We're seeing it, we're going to talk about avian influenza just for a few minutes at the end, just to kind of put it in perspective of how I think we need to constantly be aware and constantly be looking at the infective microbiome, if you will, the organisms, the subset of microorganisms that can actually lead to disease in humans. And it is surprising when one thinks about it, particularly in the area of viruses, where, in fact, over the last couple of decades, there have been several new viral types that have entered humans and caused disease. So there's something going on, and we're going to talk about evolution and selection a bit, because that's a particular area that I'm interested in for malaria. Okay, so a globalization of risk is something which I think few people thought about in detail until the Sars epidemic, at least few people in the general public. And this is just, SARS appeared somewhere in, China, Southeast Asia, but it spread extremely rapidly throughout the world, so that there was, in fact, and I didn't mean to leave South America out, this really spread, and so that there were cases of SARS every place in the world. And this is something which has changed over the past 50 years, the level of international travel. I have a field site in Senegal, and so in the winter, I always go to the Senegal field site, and I almost always have a cold or a little bit of influenza, right? I get on a plane in Boston, I fly to New York, I spread whatever I have around New York, and then I fly to Senegal and I introduce whatever it is I'm carrying into Senegal and vice versa. So that air travel or commerce has really made a difference. And I'm going to show you an interesting piece of data for malaria, where in fact we had never realized this, but it turns out that even sort of 50 years ago, an important part of spreading drug resistance in malaria is actually due to travel, not to selection at individual sites. That one resistant mutant appeared and it has essentially spread around the world, or at least around half the world. Something that I think many of us in microbiology haven't focused on. We really think about selecting changed organisms in sight, but in fact spread is probably far more important and probably has some public health implications, but we'll come back to that. Okay, this is just to depict resistance. This is now the disease that I focus on. This is from the cover of Wallace Peters' book on the chemotherapy of malaria, and this depicts resistance to the anti-malarial drug chloroquine. Chloroquine is based on the herbal medicine called quinine, derived from the bark of the chicona tree. So it's a synthetic derivative. It was probably sort of developed in the late 1930s, really introduced heavily into heavy use all throughout the world, but particularly in Southeast Asia and in South America in the late 1930s, early 1940s. And it took about 20 years for resistance to appear, for resistant organisms to be selected. In fact, for those of you who know about genetics, the Brazilians did an experiment. I don't think they intended to do an experiment, but they decided chloroquine was such a good drug and distributing it was so difficult, in a medical sense, so they thought, why not just put it in table salt? And so they actually supplemented table salt with chloroquine. And for those of you who study genetics, that's exactly the way to select a mutant. You put low-level drug pressure on an entire population, and what happened, of course? So there have been 2 chloroquine-resistant occurrences, one in South America and one in Southeast Asia. And the rest of the resistance in the world is the spread of these 2 resistant genes throughout the population. It's an actual change in a parasite gene, so it's not like resistance in bacteria where it comes in a plasmid, but in fact there's a change in the parasite genome, a set of mutations, and that region, that chromosome, has essentially traveled, and in fact the Southeast Asian one was much more active in travel. So in fact, the Southeast Asian parasite populates all of Africa. The South American parasite really stayed in South America and for some reason Papua New Guinea. So there must have been some transit route between Papua New Guinea and South America. And we can actually follow historically the resistance moving throughout the populations. So what this says, and I'll come back to this when we talk about selection, but what this says is that a successful mutation has to be not only successful in having resistance to the drug, but it also has to be successful in competing with all of the other malaria parasites, all of the other microorganisms that are out there in the world to survive, both in the presence and absence of drug. Okay, so another little quiz. If I tell you that 1%, I'll write it down, I won't tell you the answer until later, but if you have a 1% parasitemia, and I'll tell you what that is in a minute, on average in the world, and there are 500 million people infected with malaria each year, let's figure out how many parasites there are in the world each year. We'll come back to that. Okay, so one of the problems that we face in malaria is this emergence of resistance. So this is just a timeline to show you. That's chloroquine. That turns out to have been the absolute best antimalarial drug ever developed. This is Fansidar. This is a combination drug that targets nucleic acid synthesis. Resistance appeared very rapidly. This is mefloquine, another drug based on quinine. But here's the winner. This is atovaquone. This is the last drug that was developed by the major pharmaceutical industry for malaria. So this issue, these are diseases that aren't important to us. It failed as a single drug in the clinical trials. So people were being treated with this new drug. They failed, and the protocol calls for them to be retreated, right? And so a parasite that was 10,000-fold resistant to the drug was isolated from a single individual. So of course, this drug was never released. It's a single point mutation in a single mitochondrial gene, the cytochrome B gene, which leads to full level resistance. So of course, this drug was never released as a single drug, but it is, if those of you who've traveled in malaria endemic areas may have taken it, it's part of the combination drug called Malarone. And we anticipate once this is released for general treatment, we will see lots of resistance. So that's, of course, one of the challenges, and we'll come back to talking about that later. I think this speaks to the issue of the investment in infectious diseases. This is just of the 1,393 new chemical entities that were developed into drugs over a 25-year period. I think this period ended in 2003. A very small slice. You can barely see this slice, right? It's less than 1% were for any of the diseases that are important in the developing world, including tuberculosis. So this is, part of the issues surrounding the fact that these diseases are still a problem is related to, as someone has already pointed out, the lack of investment. And this is just some data to support that. Okay, so I just want to talk a little bit about sort of other health threats. So this is to remind me to talk to you about avian influenza. We're going to come back to avian influenza at the very end. But again, this is an example where this is occurring in real time. This isn't a historical issue. In fact, all birds carry influenza virus. But occasionally, there's a set of mutations, a set of combinations of genes and changes in those genes which cause a jump from this host, which is the natural host for the virus, into alternative hosts, which can include humans. And that can lead to epidemics, and we'll see some history of that. So it's important that this is not just looking historically, but in fact looking forward. All right, so let's talk about some other diseases. What do these individuals and this individual have in common? You've had the tour of the yard? That's John Harvard. Yeah. What?
Yeah,
The
it was
model.
a model of John Harvard.
It was a model of John Harvard, yes. But what did John Harvard and the people in this, what infectious disease did they share?
Consumption?
Consumption. Yeah, that's a good answer, right? Yeah, it's tuberculosis. So all of these people, including John Harvard, suffered from tuberculosis. It's a very major disease. You saw that on that second slide. But TB rates in the world are rising. In part, this is associated with the HIV/AIDS epidemic. In part, it's associated with multi-drug resistance. In part, it's associated with increased travel, changes in living conditions where you have increased transmission. It's the most common cause of death in HIV, particularly in Africa. About a third of the world's population is actually infected with tuberculosis. There are about 8 million new cases each year and about 2 million deaths, and drug resistance is rapidly rising. So again, this is a very major problem, and one which is, of course, in some ways a kind of different challenge. Here a third of the world's population is infected, yet only a relatively few people are active cases. So there's this huge reservoir of people who are infected. I would guess, if we went around this room, if this were an international audience, probably 15 or 20% of the people in the room would be PPD positive or would show signs of having been infected with TB. So one of the challenges with this disease is, in fact, it's not only the acute infection, but it's that huge reservoir of people who are carrying bugs in a latent or persistent phase, and that's one of the big research challenges. We don't understand why that happens, and we also, when the virus or the bacteria in this case is in that state, it's not very susceptible to drug. It's not as if, because it's not replicating, it's not growing, and most drugs target organisms that are reproducing. So it represents a sort of different kind of challenge, but an equal kind of public health challenge. Okay. All right, so now we're going to come back to talk about malaria. So this is one of the key players in malaria. This is an anopheline mosquito taking a blood meal. And here are the parasites. This is a scanning electron micrograph colorized to show the parasites in their majority space in the world. They live inside red cells, so these are red cells, and these are infected red cells. And you can see this red cell has burst open, releasing the parasites to go on and invade new red cells. Okay, and this is a true parasite. It uses hemoglobin, the sort of cytoplasmic content of the red cell, as its source of amino acids. It only has one essential amino acid, and that's an amino acid not found in hemoglobin. So it's a very, sort of, in terms of green organisms, this is a very green organism. It clears out the space in the red cell to be able to grow and uses the hemoglobin as its source of nutrient. Okay, so this, just to remind me to tell you about the life cycle, this is a relatively complicated life cycle for infectious organisms. The parasitic organisms, both the protozoa, of which this is a member, and the helminthic or multicellular parasites, have the most complex life cycles of infectious agents. So it's just important to understand the biology and the complexity to think a little bit about it. So this is the way I like to think about it. It's a bit human-centric, perhaps. Since the mosquito is actually the definitive host, it's where all the action occurs. But nonetheless, this is how we've drawn it. So if an infected mosquito bites a person, they inject the parasite. So the parasite actually multiplies or resides in the salivary glands of the mosquito. And as the mosquito injects saliva, it also injects parasites. And it injects saliva to keep the blood from coagulating as it feeds. And so infection is kind of just an accident of mosquito feeding. The parasite then goes to the liver, and there it infects liver cells, and it can either multiply to many more parasites, and actually it's fascinating, it can go from 1 parasite to 10 to the 4th parasites over a period, in the case of Plasmodium falciparum, the major human malaria, over the case of a few days, of 4 or 5 days. In other forms of human malaria, the parasite enters, but then it goes into this kind of, it's called a hypnozoid, a hypnotic or sleeping state. And so people who are infected with that organism can have recurrences of disease without reinfection after many years. But for the most part, the deaths and the important disease from the point of view of transmission in the world is still Plasmodium falciparum. So it goes through, it multiplies, and then these small merozoite forms of the parasite come out and invade red cells. And this is the part of the cycle. And then the parasite grows inside the red cell, chews up the hemoglobin, multiplies, and in fact then bursts out and reinvades. And this cycle, okay, is what causes all the disease. This is how you get sick from malaria. This is where most drugs are targeted. But of course, this is a dead end from the parasite's point of view. The important part of the cycle from the parasite's point of view, if parasites can have a point of view, is the sexual stage, where it actually differentiates into the female and male gametes, or gametocytes and then gametes. These are taken up by the mosquito, and this is where fertilization occurs, sex occurs, these organisms exchange DNA, and then they grow in the mosquito. So that, in fact, this whole cycle, all parasites, all malaria parasites that infect humans are either in people or in mosquitoes. There's no animal reservoir, there's no place for them to hide. They have to either be actively growing in the human or they have to be in a mosquito. So it's surprising, you know, you think about this, why haven't we gotten rid of this disease, right? Yeah, question?
It's just something I thought of real quick. Is there any important ecological roles of mosquitoes? Of course, I'm sure there are, but like, we spray the, like, what do they?
Oh yeah, what use do mosquitoes have in the world? Well, yeah, I mean, you know, so mosquitoes, all organisms have some use, right? I mean, certainly in the case of the mosquitoes that cause malaria, they have a very firm ecological hold in Africa. Anopheles gambia is a very strong transmitter of disease. It's interesting that in the natural state, we actually think that resistance or reduced transmission by mosquitoes is the natural state of mosquitoes, that high transmission is actually slightly deleterious to mosquitoes. So it's a good question. I don't know the answer. I often ask myself, you know, if being infected with the malaria parasite, because, you know, their salivary glands are filled with these sporozoites, at least for experimental infections, and you would think that would reduce their ability to blood feed, but nonetheless, transmission continues, and in fact, in many places where there's high transmission, you actually find mosquito subspecies or variants that actually are able to transmit very efficiently. So there must be some selective advantage to the mosquito to be carrying malaria parasites. And no one knows what it is. It's a great question because, of course, there are many interventions aimed at trying to interrupt this transmission between humans and mosquitoes, and a reasonable amount of effort devoted to trying to engineer mosquitoes so they no longer transmit the disease. But we don't understand the drivers of why disease transmission. I mean, I'm often, you know, shocked at how efficient this is in the world. I mean, there's more malaria in Africa today than there was at the time of the last malaria eradication program in 1950. I mean, this is a very successful parasite, and we'll talk about success, and we'll talk about, starting to get some idea of what's selecting the parasite, but I don't think we have a good sense of what's selecting the mosquito. But it's a great question. What good are mosquitoes? They're there to, destroy picnics. Okay, all right. So now we just talked a little bit about selection on the mosquito, but in fact, we have very good evidence that the parasite is selective on the human host. And this just shows you the distribution of falciparum malaria and the distribution of sickle cell anemia. And I think most people in the field of human genetics feel that the selective force on the sickle hemoglobin trait is in fact due to malaria. It's coincidental evidence. When the parasite grows in a red cell that contains sickle hemoglobin, it cannot replicate. And so it's sort of a selection on the human population to favor a trait which protects people from severe malaria infection. And in many parts of Africa, particularly in this kind of sub-Saharan African band, the percentage or the penetrance of this trait is 25% in the human population. Now, because it is a homozygous lethal in this environment, that's as high as this trait can go. So what that means is that the malaria parasite is selecting human populations at the same level as homozygous sickle trait. It's a balancing polymorphism. And it means, it does indicate to us that malaria is driving, as a very strong driving or selective force in the human population. And for many human geneticists, they feel that this is actually the strongest selection they've ever seen in the human population. That survival of malaria, and obviously this is before drug treatment, before anything. So this is just survival it's a trait which promotes survival. So I think that's a very strong piece of data. And clearly, as we're doing the Human Genome Project, and I'll talk about that a little bit in a moment, we're finding many more traits that are favorable in favor of the human for protecting them against malaria, but they're not as strong as this sickle hemoglobin trait or the Duffy receptor trait. Okay, so now we're going to talk a little bit about drugs. So this is just a little bit of history. I mentioned chloroquine, and this is quinine, the bark of the chinchona tree. It was actually discovered by Jesuit priests, and Oliver Cromwell died from malaria because he apparently, either his physicians or he refused to be treated with the Jesuit bark. And so, this is apparently a true story. So this is, the Jesuits discovered it, and it was just used as an extract of bark to treat fever, but it is the basis essentially for all modern anti-malarial drugs. So it's a very interesting sort of sense of discovery of something in an ethnopharmacopoeia way, in a sort of natural folk medicine way. The newest of the anti-malarial drugs actually was discovered in China. It's the Artemisinin derivatives. It's a derived product from a plant, Artemisinin annua, and it was used in China for about 2,000 years for treatment of fever, and it turns out to be the basis for the most active of our recent antimalarials.
Question?
Yep.
Have they looked at different varieties of the chinchona to see if there might be, as the malaria has changed its resistance, see if the tree also has different?
Well, there's a really active effort in, generally speaking, in looking for natural product antimicrobials, right? So that quinine or quinine-like molecules have been discovered many times, but generally speaking, that chemical structure leads to the same kind of mechanism of action. And there isn't much variation in chemical structure, unlike genetics. So it's, the chemical structures have fewer opportunities for variation. So, one doesn't know exactly why many of these evolve, but certainly some of them evolve as antimicrobial protections in the sort of ecological world. Right, okay. All right, just to remind me to tell you that we had transmission of malaria in the United States through the 1970s. And these are the areas where there were Anopheline mosquitoes capable of transmitting. The last transmission was in the Tennessee Valley. And how many people know how we got rid of malaria in the United States? So we no longer, except for episodic transmission, almost every year there's transmission of malaria in the United States, but it's usually from a traveler who comes back, has malaria, is bitten by a mosquito that can carry the disease, and then there are cases that occur around that. There are about 1,500 cases.
Well, I don't know overall, but I actually, this kind of point of interest, I understand that there's a swamp. There used to be a swamp in Fresh Pond area, and that was filled in at the turn of this last century because of malaria.
Right, there was transmission in Boston, yeah.
That was, I think, one thing, filling in the swamps.
Right, so there's a lot of sort of ecological, environmental control, yes?
Isn't there a big effort to spray with, I don't know if it's dioxin or what it was, that covered and presented an oil covering over the pond?
Right, yes, there was a lot of oil used on the surface of water. So that was just straight oil, right? And then there's residual insecticide, yeah? You want to?
Oh, it's just pesticides.
Yeah, so residual insecticides, right. And what else? What else happened? I bet you most people don't know this, but we'll see. What else happened?
Would it be a weather-related thing that,
Climate?
severe climate that in the years?
No, there's probably not, use of screens, okay, so it's sort of environmental. People lived inside, so they came into less contact.
Quinine and tonic water?
Yeah, so there was good case detection and treatment, right? Not quinine and tonic water, but good detection and treatment.
Does it have to do with the type of mosquito? There's a different...
Well, in the United States, it's not a very good vector. So, it wasn't such a tough job to get rid of the mosquito or to change the mosquito's habits. Well, okay, so it in fact, it's the main component probably of our success. All of these things played a major role, but the main component was that there was actually a program developed to control malaria in the United States, and it is actually the precursor of the CDC. So it is that there was a central kind of management of all these different kinds of interventions to prevent and control disease. And so CDC that was the first time there were reportable diseases in the United States, and this was the first reportable disease. And in fact, they traced every single case and did a case detection until basically there are none. So today, if a case of malaria is reported, the CDC sends out sort of the equivalent of a CDC SWAT team, and they figure out where that came from, and they control it. So, 2 years ago, there were half a dozen cases in Virginia of malaria, and they had no idea. Usually they can identify the traveler and the travel history, so it's not very complicated. But they never found the incident case. But they had people all with the same, and now we can investigate genomes, so we know about transmission. Okay, all right. All right, so now I'm going to just switch a little bit to talk about kind of modern approaches and work that I've been directly involved in, and I'll be mindful of the time, because once I start talking about my own work, I'll apologize in advance. I get excited, and then I forget that not everyone has exactly the same level of interest. Okay, so one of the most exciting things that's happened from a research standpoint is that we now really have a whole new set of tools to begin to understand these organisms. And I think someone said it at the beginning, this just hasn't been a focus of research. And so, yet they're fascinating organisms from a biology standpoint. Here's this organism, has the genome about the size of a baker's yeast, and yet, it goes in liver cells, it goes in blood cells, it's in mosquitoes, it's survived all of the selection of the human immune system, and so it's fascinating. And having the tool of having the genome, so the sort of one of the spin-offs of the Human Genome Project has been that the technology to look at the genomes of all sorts of organisms has really exploded. And the malaria parasite genome is about 1% the size of the human genome. So it's almost like a toy genome. when I went to the Broad Institute on sabbatical, and I'm going to tell you about some of that work, and as I started telling them about malaria, they kept saying to me, but what size of the genome? And I'd tell them, and everyone in the room would kind of chuckle and say, no, that can't be right. This is a malaria biologist. She doesn't understand genomes. There must be a mistake. And so, finally, I gave them the paper that showed them the size of the genome. And they said, but this is 1% of the human genome. So they realized that a lot of the tools they developed for the human genome, so for example, the analysis of the parasite genomes can be run on a computer that size, whereas, and in a short time, so that they now actually use the malaria genome to train some of their software for the Human Genome Project because it's a much smaller genome. So we have now the complete sequence of both the Plasmodium falciparum and actually just recently published the Plasmodium vivax genome, the other major human malaria. We have the complete genome of the Anopheline mosquitoes now. So some of the questions you're asking about what are the selective forces on the mosquitoes, I think we'll begin to be able to ask and answer. But I'm going to concentrate on the parasite genome for today. So that, of course, opened up the field. And then, as I said, as Eric Lander formed the Broad Institute, he invited John Clardy and I to come and help direct the Broad Institute's interest in infectious diseases. And I said, well, if I'm going to do this, I need to come on sabbatical and spend time really understanding how genomics, and the overall vision of the Broad Institute is to apply genomic science to important medical problems. But in order to do that, I felt I had to understand genomic science. And so I went there on sabbatical in January of 2005 and spent about a year there sort of beginning to learn about genomes. And the result was, of course, this project looking at diversity in Plasmodium falciparum, which is now a collaboration between my lab and Eric's lab with collaborators both around the Broad and then internationally. And I'll say a little bit more about this later. And so what we set out to do was actually look at parasites from all over the world. And because the sequencing technology had changed so much, we sequenced, at least in these initial experiments, about 20 different parasite strains from across the world to ask, and we picked parasites parasites derived from different geographic locations with different phenotypes, resistant to drug, carried by different mosquitoes, to get as much diversity to kind of describe the population overall. And then we did what's called single nucleotide polymorphism discovery. So we completely sequenced, and to different, sort of degrees of coverage or the number of times we sequenced the genome. We sequenced two very interesting drug resistant. This is a highly drug resistant strain. This is a highly drug sensitive strain, so we'd be able to look for differences in drug resistance. And then we did a much sort of more just sampling of another set of strains and discovered about 100,000 single nucleotide differences. So these are changes in the DNA sequence from one parasite to another. Just as if you sequenced your DNA and my DNA, we'd have at various positions differences in our sequence, some of which determine the difference in our traits. Okay. And so the idea here is to just look for the differences and then see what those differences tell us. And so we've gone on, there are other groups working on this, so there are about 100,000 of these. And so what we're really asking is the following question. If we look at the parasites today, so what I call the extant or the current population of parasites, you have to kind of think of these parasites as survivors, right? And this is the history of, sort of the parasite genome. It started, we can trace, in this example, we can trace the parasite back to its primate precursor. This is a parasite called Plasmodium reichenowi, which grows only in Old World primates. And then, clearly, as we were talking about, and I mentioned sort of in the beginning about this notion that at some point this parasite had to jump from the primate into humans. And then it had to kind of survive in humans. And we know from looking at the history of the populations, or at least we think we know, that there was probably some really severe bottleneck as the parasite went from primates to humans around 100,000 years ago. But then, around 5,000 years ago, there was another very severe bottleneck in which one or a very small number of parasites survived. And at this time, this is the time that people began living together. So this is the time of agriculture, so bigger communities. And if you think about it, a parasite that can only grow in humans, right, has a big advantage if there's a bigger human population. In the hunter-gatherer environment, there were a few people, they'd get malaria, they either survive or die, but from the parasite's point of view, it was kind of a dead end, right? Whereas once you've got communities of people living together, you can actually set up kind of transmission, and you can have epidemics, and then it can become endemic, where, you know, from the parasite's perspective, survival is an advantage. And clearly, and we also think this is the time where the mosquito, the Anopheles mosquito, began to be the primary vector, so that both people living together and a vector coming together, because people now accumulated water, and that's the convenient place for mosquitoes to breed. Since then, clearly, and we're fairly sure that the origin of Plasmodium is in Africa, but then there was dispersion to continents, and then there are all sorts of selective pressures on the parasite, including the one I mentioned earlier, that is drug resistance. Or drug pressure. We've had drugs in the population kind of widespread for the last 50 or 70 years. So we expected when we began this work that in fact this would be the major selective force on the parasite. Yep.
So I'm curious, in B, the bottleneck is caused kind of by a crisis, but in A, is bottleneck caused by an opportunity?
Yeah, exactly, an opportunity. Yeah, sort of, it's probably the expansion of some organism that was transmitted best, right? You can imagine that something that got into the mosquito best and then got out of the mosquito into people would quickly outgrow the population. And that's why we say it's probably not a single one, it's probably a few genomes, but right. Whereas here, it's probably a very strong selection of something which can go from growing in the primate to growing in the human. So for example, we can grow the malaria parasite in primate red cells, but it doesn't do very well. You know, it kind of ekes along, and eventually we can't keep it going continuously, whereas in human cells, we can keep the parasite growing forever. So there's something that happened. We don't know what that is. And of course, these kinds of studies may give us a hint. But so we went into this really, my whole mind was around drugs, right? And drugs. And so I figured the real footprint of selection would in fact be selection by drugs, and I'll show you in a minute. But what did I forget in thinking about this? What's another major selective force on a microorganism? Yeah.
Well, you talked about how it's need for water for some of its transmission for it to lay its eggs.
Okay, so for the mosquito, right, there's strong selection on water, and also mosquitoes have selection on the parasite. Okay, yeah?
I think fungus and antibiotic type products.
So natural products in the mosquito, okay, good. This is all right. That's an interesting, yeah? Temperature, okay, on the mosquito. But think about the parasite now, yeah?
That sickle cell?
Okay, sickle cell is an example of the parasite sort of kind of force, so there, the example there is that's the parasite selecting the human, right? Because the human survives. A a person who carries the sickle hemoglobin gene is a heterozygote, survives severe malaria, sort of more frequently than someone who doesn't have it. And so that person or that gene increases in the population. So think about in the other direction. Yeah?
The parasite kills its host.
Okay, the parasite kills its host, so that's the sickle cell. But what's the host doing to, yeah, okay.
Well, just the host is in a way selecting for parasites that don't kill it too fast, because then otherwise it can't be transmitted, but...
Right, okay, so the host is, well, you're right, the parasite would prefer not to be killed too quickly, if as I say, if parasites could have preferences, but the host, right? What does the host want to do? Survive. The host wants to survive. So what do you have that makes you survive every infection?
Immune system.
The immune system, right. And so this was like staring me in the face, but of course I didn't think about it until I looked at the data. So as we looked at the data, right, this is now, and I don't expect you to sort of read these individual categories, but this is just a way of looking at the data. I mean, one of the things you have to learn when you work with whole genomes, there's 24 million base pairs, there's 100,000 SNPs. You got to organize it kind of into diagrams because otherwise you'll never understand it. So these are called gene ontology functions. These are functions, these are categories that are focused around genes that have sort of a common function. And the way this is arranged is the percentage of genes in red shows the highly polymorphic genes, and the order of this is actually in the order of most polymorphic gene ontology category to least polymorphic. And then this is the percentage of genes within this category that are polymorphic, or in the case of black, completely non-polymorphic or identical throughout the world. All right, all of the targets of drugs are down here. They're relatively non-polymorphic genes. Many of the drug targets, well, they show a little bit of polymorphism if there's resistance because there's a resistant mutation, but genes closely related are completely monomorphic. The highly polymorphic genes are all genes that are expressed on the host cell plasma membrane, the surface of the parasite, involved in invasion of cells, immune evasion, cytoadherence, sort of all things that are involved or targets of the immune system. And another way to look at this data, okay, so molecules associated with the cell surface are highly polymorphic compared to molecules involved in basic metabolism. The other very fascinating thing about this is these genes are completely monomorphic. No matter where we look in the world, the sequence, not just the amino acids, but the sequence of the DNA is identical, probably indicating very recent divergence, but it also may indicate selection on sequence. Okay. So this is just another way to look at the data. This is just plotting, essentially measuring the diversity. This is called nucleotide diversity. So this is counting the number of SNPs in a gene and normalizing for the size of the gene. Okay, so most of the genes in the organism are in fact quite low diversity, but there's this very long tail, which you see in relatively few organisms that have been examined to date. And these are the current vaccine candidates for Plasmodium falciparum vaccines, and they're all in the high diversity category, indicating that antigens, or things that are targets of the immune system. So what this is telling us, I think, perhaps worrisome for those trying to develop a vaccine, is that the parasite is under enormous pressure from the immune system, and you get variants popping out all the time. Now, what this implies, so what other organism is like this? What other organism that you can think of has kind of a similar feature?
HIV.
HIV is like this, right? Okay. What?
TB.
TB doesn't have such a large amount of polymorphism in it, and that's probably because of this latency, right? The flu. Influenza, right? So the classic example is influenza. Every year, I assume all of you get a flu vaccine, and every year you need to get a flu vaccine because every year the flu has mutated, has been selected for a mutant form. And that's, we think, what's happening here. And you're just looking over time. Because we're looking at a large number of parasite strains, we're sort of looking over time at what's happened since the last time they were all a single parasite. So what this implies, perhaps like flu vaccine, is when an entire population is immunized with the circumsporozoite antigen, which is the vaccine that's going to be tried in 16,000 people, we predict that certain people will have parasites that will not be recognized by the vaccine, and that it may be very much more important to think about this as influenza rather than as sort of measles or something which has relatively little variation. Okay, so that was a little bit of a surprise. So now, yep, go ahead.
So what you showed before is kind of a snapshot. So if this is occurring, do you see certain SNPs going up and other ones going down, and then a year later a different one?
Exactly. So if you look now temporally, right, you find that there's also variation temporally. And that's actually a fascinating question, what we're working on now. So we actually have parasites from probably 40 years ago through now that we can examine at certain of these sites, and we're just in the middle of that. But even within a season, in our field site in Senegal, we can see some change in the frequency in the populations, and that's consistent probably with both immune response and also perhaps with transmission. Yes?
Does this antigen diversity, in essence, then have the effect of kind of wearing down the immune system and sort of diminishing what might be an initially good response?
Exactly. I mean, I think that's, so I'm not an immunologist, and there's a lot of debate about this, but I actually think what's happening is that what you see when so if you start with a small child in disease endemic area, they're first at early birth, right after birth, they're protected because they're breastfed, and they have maternal antibody. At about 6 months, when they're weaned or when maternal antibody never doesn't play as much a role, they start getting infected and with serious bouts of disease. And all severe disease occurs in young children. So the 1 million deaths due to malaria each year are almost exclusively in young children in Africa. But as they survive that, right, by the time in most endemic areas they're 5 or 6 years old, and certainly by the time they're 10, they no longer die of severe disease. They still get parasitemic, but they don't get sick. So adults living in endemic areas carry parasites around. That's why there are 500 million. Most of these are asymptomatic, right? And that's what I said. Remember I said earlier that drug-resistant parasites have to be able to survive compete with non-resistant parasites in the absence of drug. It's because most of the parasites aren't being treated with drug. So that what I think has happened to that child is they've received sort of multiple antigenic types, and they finally now have an immune response that is able to control most of what they see. And the classic story is, of course, when people travel away, it's most common for schooling, travel away out of the endemic site, and then move back after a year or two, they again are susceptible. I would hypothesize because there is a new antigenic type. It's moved on like the flu virus. And so we're going to test that. But that is what I think is going on. There are immunologists who feel that it is much more related to presentation of antigen and variety of antigen, and they may also be right. But it's a very important sort of research point at the moment. And of course, it's very important from the point of view of vaccine design, because if you think about it, if it's a moving target, then your vaccine has to be a moving vaccine. If it's administration, just better administration of the magic bullet target, then that's what you should work on. So it has important implications for what we do next in vaccines. And at least this work is beginning to cause people to think a little bit about it. Oh yeah, I forgot to point this out. This is the most diverse human gene. It's not exactly fair because this is a diploid organism, the calculation's different, but this is its relative spot. So the most diverse human gene, focused actually on the diversification in your immune system, is actually less diverse than these parasite genes. Okay, all right, so this isn't, this is sort of not just of academic interest, but it's of course also of practical interest. We've already talked about chloroquine resistance spreading, but from a public health standpoint, selection on the parasite and how, whatever the trait is, whether it's escape from a vaccine or it's escape from a drug, whether that occurs independently in multiple sites or whether it occurs once and then is spread, is actually quite a different public health problem. Think about it. What would you do if this were, we could call this a vaccine. Let's say that, you got an effective vaccine and then all of a sudden a parasite escaped, right? And this were actually the mode of action. What kind of public health approach would you take versus independent occurrences of either drug resistance or vaccine escape?
Well, if you had the one thing, then one vaccine or one drug would take care of it. But if you have multiple, it has one particular vaccine would not be very useful.
Right. Okay. So that's right. That's a good idea. But let's say that this is already escaped from a public health standpoint. What does that mean is most important?
To track it.
To track it. Right. Surveillance. Yeah. That surveillance is now critically important. You know, we don't do any surveillance of malaria in the world. We don't track it at all from we don't look and see if the genomes are moving around. So this is actually, and if you think about it, we didn't do it until very recently for flu, and I think the Sars epidemic kind of forced us to do that. We're not doing it for HIV. We don't know, how HIV is moving around, and I actually think this is very important. Now, the reason, so let's just take a look at this. This is kind of the diagram. In this case, it's for drug resistance because we have the most kind of data on this. But I think the same thing could happen at vaccines. If in a population, before you apply the pressure, in this case it's going to be drug, here's the locus that actually encodes drug resistance. But in the population, there are lots of different chromosomes. There's no drug. The diversity along this whole region of the chromosome is really, sort of an average diversity. Once you've selected with drug, not only do you select this locus, but you drag with it all the attached DNA, right? I mean, initially you drag the entire chromosome. So the parasite has 14 chromosomes. You drag along one of those chromosomes, and that confers the trait. And then eventually, by recombination, that linkage breaks down, okay? But recent selections, actually, you can detect simply by looking at this nucleotide diversity measurement across the positions on the chromosome. And you'll find this really low diversity. And that, in this case, if you compare resistant and sensitive parasites, you can actually just simply identify a resistant locus by looking for this selective sleep or this decrease in diversity. So this we have done. And in fact, this is the diversity around the PFCRT locus. This is the target of, this is the mechanism or the gene that confers resistance to chloroquine. And you can see this reduced diversity around this gene. And this is statistically significant comparing sensitive and resistant parasites. You also see, as you look along the chromosome, that you begin to see other regions of low diversity, not necessarily associated with resistance. And of course, that may mean we're selecting on a different trait. So this kind of approach can be applied to almost any trait that we look at, that we can measure in the parasite. And so we think this is a way to identify that recent bottleneck, right? Okay, so we can now look genome-wide, and all we have to do is sort the parasites into phenotypes and then ask, are there regions of the chromosome that show this reduced diversity consistent with that region of the chromosome being selected? And then obviously we have to go in and test those genes. So it's a way of identifying the regions that we may be interested in. Okay, so that kind of is kind of a nutshell of the kinds of things we're doing, and I see we are running a bit short of time, so I'll finish. We clearly want to be able to, so we've discovered SNPs. We now have technology that we're able to actually genotype the SNPs. So we know what the SNPs are, and so we have kind of neat technology that allows us to look at large numbers of parasites without having to resequence. This is the Affymetrix DNA chip technology, and we now have a SNP chip for Plasmodium. This is the same technology that's used for human genotyping, and I can report that it works very well. But the idea is we obviously want to understand the parasite populations, the relationships of the parasites to one another. We've already talked about drug resistance and the vaccine applications of this, and we're now collaborating with the group that's testing the circumsporozoite vaccine to see if we can begin to see population shifts. We also are able, because of all this knowledge, it's interesting. So we have, we know everything about the genomes of these parasites that we've sequenced, but actually it turns out from a practical standpoint, you can apply all that knowledge, you can focus it all down on a relatively small number of SNPs and make a molecular barcode which uniquely identifies each and every organism. So it's actually like a fingerprint for the parasite. So let me just show you one application of that. So often when you're treating patients, right, or you're doing a drug trial, what happens is you treat the patient with drug, right, and the parasitemia goes down, and then you get recrudescence, right? You get parasites. But of course, we don't know, without having a unique fingerprint for this parasite, whether that's the same parasite and it's actually resistant or it's failing whatever the treatment is, or whether that actually is a reinfection. Remember, these people live in areas where often you can get 300-400 infective bites a year. So over the course of a drug treatment trial, you may very well have somebody who's been reinfected. And so now we can really very quickly, just in an instantaneous way, tell whether it's resistance, which is extremely important, so going to this surveillance issue, or whether in fact it's reinfection and the first treatment was effective. Okay, so I think that's probably that. The other thing I would say is these are technologies that clearly are very high tech in their development, but then once we really get them to the level where they can be used in the field, we are really dedicated to being able to train scientists in disease endemic countries. And so actually, as we published this paper and made these first discoveries, we joined our collaborators at Czech Antioch University in holding a West African-wide training workshop in the use of genomic technologies in discovery, in vaccine and drug discovery in Africa. Okay, so I think with that, I will stop. I was going to come back a little bit to influenza virus, but I think I've run out of time, so yeah.
I mean, it seems like the emphasis has been getting rid of the mosquito. Now we're talking about trying to get rid of the parasite. I mean, is there a tension there in terms of what the best policy is, or is it better to have a combination?
I think it's going to be better to have a combination. You know, an epidemiological term that one should keep in mind is the basic reproductive rate of this infection is 100. So one case can lead to 100, and that's because it's vector-borne and you get multiplication both in the hosts and in the vector. And so, most diseases like smallpox or polio that have been contained have basic reproductive rates in the single digits. And so we think we'll need everything. The Gates Foundation has declared malaria eradication as their goal. Now this happened once before in 1950. The World Health Organization declared malaria eradication as the goal. And actually, I don't know if it's here. Yeah, these are we'll just skip over a few of these slides. Whoop. These are the postage stamps. I have the originals of these stamps hanging in my office. These are the postage stamps commemorating the malaria eradication campaign. In Nigeria, there's more malaria today than there was at the time of the eradication campaign, partly because of population growth. But for all of the countries, I think on this map, maybe Afghanistan has less malaria. So in general, Vietnam has less malaria. But in general, the eradication campaign was not successful. And part of it was that once, the classic example is Sri Lanka. In Sri Lanka, the eradication campaign was extremely successful. They were down to less than 20 reported cases of malaria infection. So the government said, problem solved. Stopped residual insecticide spraying, stopped case detection. And so when we were there in 1985 to begin to look at drug resistance in that part of the world, there were over 1 million cases in Sri Lanka. So and we don't know because they didn't keep parasites. They didn't do surveillance, right? So we don't have even microscope slides from that time. So we don't know if it was a successful parasite that grew out, or it was introduction of a parasite from India or from Thailand, because of course they had a lot of commercial exchange, military exchange. So again, this is a fascinating question. So we have now convinced the World Health Organization and the Gates Foundation that parasites should be collected essentially from the entire world so that we know what we're starting with, and then we can sort of map what happens. And in sites where we're actively working, we'll actually be doing surveillances over the eradication period. So in Sri Lanka, we're about to go into pre-elimination, and we've just been approved by the Ministry of Health. Nadira Kunasuriya, who was actually a Radcliffe Advanced Studies Fellow here, is chairman of parasitology at the University of Colombo, has now got an agreement with the ministry that we'll actually be able to do sequencing of parasites as they move through elimination, so that we'll actually, I think, understand in some detail what's happening. And this could be a model for other parts of the world, because it's an island, so we can actually control, at least in the sense of being able to collect data on importation versus emergence of native strains. Other questions? Yep, here maybe?
I mean, if you went out and asked the average person, what are you most worried about? I don't think malaria would be their number one. They'd say avian, flu. The avian flu. So, I mean, is there going to be some kind of advertising campaign? I mean, how do you get the money you need to research this?
It's a great question. So a lot has happened in the past, say, decade, certainly the last 5 or 6 years with active efforts. So in part because of awareness of AIDS internationally. So there's the Global Fund for Treatment of Malaria, TB, and AIDS, but really driven by the awareness of AIDS. And in the U.S., there's a reasonable amount of kind of public awareness activity. There was an American Idol show on malaria. It raised money for malaria in the spring. ExxonMobil Foundation, or Foundation that also supports some of the work we do, sponsored that. And so they took the American Idol winners. I don't usually watch American Idol, so I'm not up on it, but the winning contestants went to Angola to visit the site where they gave the money, part of the money for bed nets and for treatment. So there's a growing culture, and it's interesting, right? I mean, if you ask people in the United States, is malaria a problem? I think the answer would still be no, right? But if you ask ministers of health or people in Africa, in fact, one of the stories that Gro Brundtland, who used to be the Director General of the WHO, told frequently was when she became she was Minister of Health of Norway and then became the Director General of WHO. And during that process of getting elected to be Director General, she traveled throughout the world asking ministers of health and people within health departments what their major problem was. She expected it in Africa to be HIV, but in almost every African country it was malaria. So that I think awareness of the disease is a little bit dependent on geography. And I would say now, malaria is on a pretty high kind of level of awareness at the World Health Assembly. So African nations are asserting themselves. The Abuja Congress of the Ministers of Health of Africa have declared malaria eradication or elimination as a goal. And when we say eradication, I think it's important to realize that's aspirational. Biologically, I think it's going to be tough for exactly the reason I mentioned to you. It's just, the transmission rate is so high. But I think you'll be able to get rid of disease. If you could convert everybody into an adult living in an endemic area in terms of how sick they are, that would be a big improvement. It wouldn't be perfect, but it certainly would be a big improvement.